Okay, good afternoon to everyone. My presentation will be focused on the role of the first. I mean, the last generation of antibodies specific for therapy. So the antibodies specific for therapy of human myeloma were selected according to a very simplified approach. They initially was found something expressed by the myeloma cells, the majority of them, and that's all. So was, as I said, simplified model because as Carolina has stressed, everything is changing during the life of the myeloma. So a single target is not enough. So let me present to you the results obtained by using the first antibody approved, CD38 specific daratumumab. And the use of this antibody was approved rapidly because was inducing very good and significant results. And what appeared was interesting for two reasons. One, the antibody was doing its job as an antibody, killing, inducing, inducing signals, inducing death, inducing other things like we expect to inducing cytotoxicity, what we expect for an antibody. But unexpected was the fact that the antibody was inducing important signals on the normal cells, I mean the component of the immunomodulatory population, which are CD38 positive, and the signals implemented by these cells were inducing, were participating, were completing the effect of the antibody. So for a basic scientist like myself, the question is, how is possible to explain that an antibody is killing as expected the target tumor, but at the same time was acting on normal cells, and in these normal cells inducing positive and negative signals. So this is due to the fact that the exact effect of the antibody was not tested, it is not yet done. So what happens, and I will list rapidly with you what are the events and the elements of the immune response that take parts to the action of the antibody. The first one is an antibody is bound by a family, and a very diffuse family of Fc receptors, which are present on many of the cells of the blood. And these cells are able to induce positive signalings, but also they inducing negative signals. And also there is the last one, the minor one, the newcomer at the family, the so-called Fc receptor neonatal. Nothing to say with babies. Well, neonatal because was found in the maternal milk. And what does this neonatal receptor do to the injected antibodies? Simply, the immunoglobulin are taken inside by many cells, starting from endothelial cells. It is internalized and some way checked, goes through different pathways and may be eliminated or is re-expressed what is the meaning for the patient? This step is regulating the life in vivo of these antibodies, like happens for albumin. A third element which is important for the life of this antibody targeting CD38 is the fact that the signals mediated by CD38 are a function of different elements. CD38 has a very short cytoplasmic tail for this reason. CD38 in many cells, excluding myeloma, is located in membrane lipid microdomains hosting the most important receptor, T cell receptor, B cell receptor, and CD16 on NK cells. So this is another part of the story which has a potent role in the activation and suppression of this population. So considering this start, we asked ourselves what happens when an antibody binds the target. We exploited very old observation coming from the immunology. So when you cover a target, an antibody at 4 degrees, the target and the antibody simply stay there without moving. But if you do the same experiment, and also like happens in vivo, you have an induction of a so-called polar aggregation of the complex antigen, in this case CD38, and antibody, and this pole is induced to be released. So this is what happens, and it is a physical view what happens. This is 4 degrees, and you see there is an even distribution of the complex. The complex is shown in green. But if you work at 37 degrees, you have a polar aggregation, and you see that almost of the surface, myeloma surface, is deprived of the target of the antibody. So the question is, what happened when what happened inside? So we tested what happened in a myeloma when you treat with an antibody, in this case with daratumumab. There is a significant modification, and you see that there is an increase in the ligation of selected sequences which are related to metabolics, call it this way, and downregulation of several other sequences, some way related to cell cycle. And let's not go further. So I was presenting you the so-called polar aggregation, which becomes simply vesicles, which are expressing on their mini cells, expressing on their surface the target of the antibody, but also other ectoenzymes, components of the complement, PDL1. And the obvious question is, where do these vesicles go? These vesicles can go through the tissues, and the most important things to be remembered is that they are mini vesicles covered with an antibody. So their destination is mandatory. They go where there are Fc receptors. So This is a slide showing what happened on monocytes, which are the cells most rich in Fc receptor. And not only they accumulate on top of these cells, they enter. Now we have the complexes in red this time that enter the monocytes, but also other cells like myeloid-derived suppressor cells, which are less rich on Fc receptors, or the NK cells. I mean, we paid attention to NK cells since they are the cell population which show an apparent dichotomic behavior. They disappear quite rapidly after the injection of the antibody. So we tested what happens inside, and the inside, after the injection of the capture and engulfment of the vesicles, there is an increase of the immune response. Let's say this is a selection of things that the computer says immune response related genes, and this is as expected. I see will take a life to go through all of this, but if you are patient, you can see that some of them probably members of your study are included here. And also mitosis, I mean, genes some way related to mitosis, nuclear division. So this is a physical representation of what happens in NK cells, which are the first step are activated, but after a rapid period, I don't know, in days or hours, they disappear, probably they die. And you see all these cell cycle processes. I mean, there is an induction of death of this population. So what these are, all these aspects are not well studied in vivo, because it's difficult, because the condition of the patients are critical, and also this is not critical aspect in general. In my view, indeed are very important because the a significant portion of patients develop resistance to the antibodies. So each of these steps that I presented should be tested. So I presented here what happens using daratumumab, the first one in the row, and the most used so far. So let's see what is the difference with isatuximab. Isatuximab is the second antibody approved specific for CD38. And the first thing that is important for me is to remember that the analysis, very nice paper by Bruno Paiva, has shown that the antibody is able to trigger a cytotoxic activity only in the presence of high density of the target molecule. So in this condition, you see no effect. So this is not a defect of the antibody, is a real good quality, since the normal cell population expressing CD38 is an ubiquitarian molecule, are some way protected or unaffected. Only in the presence of myeloma or NK cells, you have activation of ADCC, phagocytosis, complement-dependent possibility, cytotoxicity, and also another step that could be considered and could explain the different effects mediated by the antibody is the fact that the myeloma model is the only, or to my knowledge, is a model where the target molecule is expressed on the tumor, which is Fc receptor negative, and on the majority of effector. So what may happen inside is the fact that beside this is the normal pathway of activation. You may have a double binding by tail and by the specific region, and this could explain the effects sometimes going in different direction. So, but the most important difference, which is for me determinant, is the fact that CD38 is has many functions, but the most important is that it is a NECT enzyme. NECT enzymes that is able to its substrate is NAD, and so in that case, NAD, you know, is an important source of energy and signals. So the obvious question in front of this property shown by CD38, what does the antibody do on the enzymatic activity? So you see here, this is a comparison between isatuximab and daratumumab, and you see that daratumumab is a quite strong inhibitor of the enzymatic activity of CD38, even though not able to reduce totally its enzymatic activity. They do recognize different epitopes. And this is important, and this is something to test in vivo. So the enzymatic activity of CD38 has something to do with the clinical effects. Don't forget that CD38 is able to produce adenosine, and we tested and we demonstrate on the side of the bone marrow niche of the antibody, which is an immunosuppressant and can contribute to immunosuppression observed on patients. And this is a summary in a recent paper which compare all the effects mediated by the two antibodies. You see here is versus DARA, ISO versus DARA, and you see this isatuximab is not a simple copy of daratumumab, and so we need some time to have a complete picture of the differences. And the problem is, if we observe, I mean, you are clinicians and you observe resistance in clinics, how can we survive this? So one possibility is to modify the structure of the antibody another to work together, other agent or suppressor. My favorite is anti-CD47. The CD47 has a very nice name, eat me not molecule. So I would like to know, and this is coming soon, what happened in vivo using these two antibodies. So the isatuximab has been modified by Angela Virone-Odos in the FC receptor, improving the ability to be captured by different receptors, even though the clinical results seems not to be satisfying. Another good one is the antibody prepared is called biparatopic antibody. There are two arms linking to different epitopes of the region of the antibody which host the enzymatic action. And so, as you can see here, there is this time a strong inhibition of the enzymatic activity. There is only a minor defect, doesn't do anything in terms of cytotoxicity and other things. So the story is not finished. I'm just showing here that there are at least 15 trials or attempt to use CD38 related sequences for the production of antibodies or other things. But the question presented by Dan Longo in these years, so 7 years ago, not yet are fully answered. I hope that the next presentation or next time we will have more details to answer this question. Thank you for the attention.